There we go. That's better. All right. So um, I can go through this step by step if you wish. Oh, dang it. Or I can go through particular bits that you struggled with. What would you prefer? It's kind of up to you. Or I could just go and have a nap. That'd be nice. Snore away in somewhere comfortable underneath a fan. Sleep off my lunch, that kind of thing. No? Yeah. Oh well. No nap for me then. So this is one of the weirder aspects of teaching online. I'll just assume that you want me to go through it all. Okay. I'm going to actually have to share my screen as such. And so really what I did is I actually just went through this myself, right? It's oh, okay. So okay, let's actually, you know what you could do to help me? Because I can't bring the questions up without showing the answers, which would kind of defeat the object of doing this. So why don't you copy and paste into the chat box what each question is in turn, and then we'll use the information that we're gathering to answer those questions, and hopefully that will kind of help make it a little bit more straightforward. Because there's no kind of trickery involved here. It's all intended to be pretty straightforward. Okay, so let's go to Flybase, which is the site of a much more inferior model organism to one that I work on, obviously. Oh, look at that. It even remembers my choice from last time. Right, so you go here, put in yellow. That just kind of did it for me. And what you'll see come up, let's get you back over here, is, no, don't want that, go away. All right, can someone else who hasn't finished the assignment yet go and, actually, Angela, do you want me to do what I proposed earlier? Yeah, we can do them one at a time. So if you want to just copy and paste question 1 into the chat box so everybody can see it. What chromosome is the yellow gene on? Awesome. Angela, it's up to you. It really is. If you're happy with what you got, that's totally cool. If you want to try it again, you're also able to do that. Yeah, exactly. So this information here is what tells you what chromosome it's on. So this is a sequence location. These are actually the individual nucleotide numbers of the gene from beginning to end, right? And so this tells you exactly where on this chromosome the yellow gene is. So the answer is obvious, right? It's highlighted. Okay, what was next on the list of things to do? Okay, so let's go and have a look at the yellow gene in detail, right? So you got to click on that icon and we'll scroll through and have a look at the summaries expansion tab. Go away. Right, so click on that. Dum, dum, dum. And then this will take us to the page where all the information available for yellow gene or any flies that have the yellow mutation in it can be found, right? So we've got, you know, the species, we've got the symbol of the gene, we've got some summaries, right? So here's some information. What's the next question, Ali? I think it's about what phenotypes are seen in yellow flies. Oh, okay, right. So At the very least, male courtship is one. Characteristic yellow color is another one. Right? Oh, that's two. Right, but I don't think that's all of it. So we want to scroll down to here. Right, so the summaries tab, and that's minimized by default. So you have to click on it to open it. And you have all of these ones here, right? So cuticle pigmentation, we know that because they're yellow. That's pretty obvious. Male courtship behavior, veined wing extension, whatever that is implies, no idea. Melanin biosynthetic processes, which kind of makes sense because melanin's a pigment. And this also tells you where it is expressed as well, right? Which will tie into essentially what its roles are. Okay, so that should help you answer question 2. All right, what's next? So now we want to go and have a look at what alleles we have. Yeah, thank you. Okay, so that should probably be under a heading saying alleles and something or other, would be my guess. Here we go. Oh, no, that was already open. So click it back again. Okay, so we have 80 pages of alleles, right? If we put it on, actually, even more fun, let's put all of them. This might take a while. See, but you never knew you could have so much fun looking through sequence information. Right, so these are all of the alleles that have been discovered for the Y gene. You don't really care about that because you just want to know which one is the most commonly studied, right? Not the one that's found most frequently, but one which is in the most number of stocks. And the winner is, with 49,414, this allele, right? Y and then 1. And that basically, in Drosophila speak, that's the Y, first mutant Y allele discovered. Y plus is wild type, if you remember from the notation that we used before. Okay, so that should tell you the answer to question 3. Okay, and we're working with the Y1 allele. So we want to know more about this, right? First off, note that it says it's a loss of function allele, right? That will come up later on in one of the other questions as one of the answers, right? So let's go and actually have a look to see what the Y1 allele is. Okay, so first up, it's a point mutation, right? So this is, if this is the whole gene in bluish, whatever color you would call that, pardon me, it tells you it's a point mutation, and point mutations are single nucleotide changes, right? So it's telling you that one nucleotide has changed between the reference or wild type sequence and the mutant sequence, right? We don't know what consequences would have right now, but we do know that it's a loss of functional allele. So it means that there is no functional protein produced, right? Okay, so what does this look like? Oh, here we go. And this is the information that you need, right? So you know that ATG is the start codon, right? What does in RNA speak anyway? I think Andrew actually wrote this in the last, or was that Gabrielle? Can't remember. What amino acid does AUG? Yeah, methionine, right? Yeah, that's right. So if you have no start codon, do you have a protein? No. You do have an mRNA, right? So this mutation does not affect transcription. So you will get a beautifully transcribed mRNA, which will have CTG, or sorry, CUG as the first codon. Now that doesn't code for methionine. I don't know what it codes for. You could look it up. And so translation will not occur because the ribosome will not recognize a start codon as being present, and it will not produce a protein from that sequence, right? So the fact that it's a loss of function is not because of truncation in the protein or lack of transcription or like a frame shift where you get like a weird ass like gobbledygook protein. It's because there's just no protein at all, right? It takes out the start codon. No start codon, no translation. Okay. What was the, I think that was the answer to the next question. Ali, what was the next question? What was the number? Question. Which are true of the following of the Y1 allele, right? So you need to work through those. In some of these answers, you're actually being asked the same thing in different ways, right? So you need to think carefully through what's going on. Right, let me just stop this so I can go back and make sure that I remember. I don't know how many of you are quick enough to see the answer, right? So you got to look through, and I'm just reading the answers here, and figure out what it is you're actually testing, right? So you know essentially that oh yeah, I mean, but thank you though for Ali for posting it up, right? We know that these flies are yellow, and we know that they're yellow because of a mutation in the yellow gene, right? So that should kind of knock out some of those hypotheses. The question that we're really asking in this particular project is what changed results in change in the yellow gene results in the yellow phenotype, right? So read through those hypotheses, those varying different hypotheses, and see which one you think best fits the, hang on, got to get off of that. Let's put this up. See which you think best fits the options that you're being given. Let's get the chat back up. Okay, so that's really just a what am I working with here kind of process, right? So now you know what kind of gene we're working with, you know what's involved in, you know what the nature of the allele is, what change has result. And yeah, that's because one of them's really right and one of them's not quite right. So you got to be very careful thinking about what exactly are you testing. Yeah, well, blame Dr. Page for that. He wrote this. I absolve myself of all responsibility. No, I don't really, but that's one of those, you have to really kind of understand what you're doing to differentiate between those two things. So that's why it's there. Okay, so what we're going to do next is start looking at the gene itself, right? So this is a kind of effort to, you know, get you through and explore essentially what a gene looks like, right? What are different functional parts of the gene, and pardon me, what do they mean, right? Kind of visually interpreting the information that you're getting. Okay, so we want to go to NCBI. We're going to get out of Flybase. We don't need that no more. And this is basically where all gene information is present. So all of fly-base and worm-base and all of that information is actually present here, but it might not be present in the same degree of detail, right? Because this is really more for just sequence information. Although PubMed also does papers as well, so that's a handy resource. So you want to type in, you can do it fancy, like Dr. Page does, or you can just do that and it'll pick out the same thing. Hit return, and just double check, make sure you do get Drosophila melanogaster up, because otherwise it will throw you off. And let's click on this. Okay, so this provides a whole bunch of information, and it's really nicely done visually here too, right? So this bit here, I'm slowly circling, kind of highlights other things, unfortunately, is the gene itself, right? So there is obviously kind of regulatory stuff kind of up there that's typically not delineated in these genome browsers, just because it's hard to know exactly where it stops, right? There could be elements like woo off in the distance. And so typically what you see in genome browsers are the extent of the mRNA, right? So what this particular picture tells me is that this gene has a 5' UTR, that's the pale green bit, has a 3' UTR, also pale green, and you can see why in a second. Let's click on that. Yeah, beautiful. Right, so this here is the, get off it. Yeah, I know. The red bit is protein. When you bring it up, it should say, got to look at all of this stuff. Let's bring that over here. Gives you the length of the protein, and you know that it's protein because, God damn it, get out of it. All right, let's see if I can point here. It's shorter than the mRNA, right? So blue, oh, okay. Yeah, so green is the gene itself. So we have UTR exon 1, intron, there's only one of them, exon 2, 3' UTR, right? And does it actually tell you that? I don't know. You're gonna kind of like wander around these things. There's so much information here that just kind of tooling around a little bit will unearth interesting stuff. And you know that this is protein because it's shorter, right? Because the untranslated regions are, as their name suggests, not translated. So we have a gene which is 4.3 kilobases long, 4,325 nucleotides. Our coding sequence, which is exon 1 and exon 2. Yep. No, don't want that. Damn it. Sometimes these things are too interactive. No, don't do that. How do I get that thing back that I had before? Oh, that'll do. Back. Give me a moment. I think my touchpad has got some like cat fur on it or something. It's being very touchy. All right, I'm going to touch anything now. So exon 1 and 2 added together, the coding sequence, which is what actually codes for protein, is 1,626 nucleotides long. And that makes sense because 3 nucleotides equals 1 amino acid. 1,600 divided by 3 is, well, essentially 540 amino acids, right? That's how long the protein is, right? So this should give you all the information you need to answer question 7. All right, what's next? I think we need to actually grab the sequence next. Yeah, there you go. Match each of following. Okay, so this is actually kind of easy just from a priori information. You know that exon 1 comes before exon 2, right? I mean, it has to, otherwise it wouldn't be called exon 1. You know that the introns are only going to be between exon 1 and exon 2 and exon 2 and exon 3 or whatever, how many exons there are. We only have one intron, so it has to be between exon 1 and exon 2. 5' UTR is always before the first exon. 3' UTR is always after the last exon. So in a simple linear fashion, you're always going to have 5' UTR, exon 1, intron, exon 2, 3' UTR. So just match those up with the numbers and that gets you the answer. Okay, so now I think we need to go to grab the actual sequence itself. Okay. Ding, ding, ding. GenBank. Where are we at? Okay, so up here, click on this. So this is essentially the all the information that you need. You can download it from one place, right? And so if you wish, you can download the protein sequences. You can download the mRNA sequences, right? And the mRNA would obviously be 5' UTR, exon 1, exon 2, 3' UTR, because we were spliced out intron 1. And so now we go here, eventually. Bum, ba-dum, bum. We bring this up. Yeah. Come on, not that far away from my router. Here we go. So here again is like all of the information and authors and yada, 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 a whole bunch of stuff, right? You don't need to know that. Just shows you how many people have worked on it. And if you want to get into the weeds, this will actually give you exon 1, for example, or 5' UTR to the end of exon 1, exon 2 to the end of 5' UTR, right? That gives you those details. So you can actually, here's the start codon, first letter of the start codon. That number will be important to the end of exon 1, beginning of exon 2 to the end, to the stop codon of exon 2, right? Hint, hint, nudge, nudge, wink, wink. And this is the actual sequence, and then sometimes they put the protein sequence underneath it. So what you want to do is you want to get this file, right? Because this is what you want to download. There we go. All right, so I'm going to download the GenBank file, which has all of this information. And I'm going to save that at some point, right? And, oh, okay. I've get out of it. I've overrun myself a little bit. So just to go back, where are we at? Right, so this is the yellow gene in NCBI that we got to. And you're going to scroll down to the genomic regions and transcripts, right? So we can look at it. And then you click on reference sequence details. Try again. Come on, touchpad. Right, that will take you to this page. Now, click on GenBank. Da-da-da. That will bring up all this information, right? You don't necessarily want to read all of that. It's not super interesting, but it does have some interesting or useful nuggets in there, right? And to actually get the file, what you're going to do is you're not going to download it, you're going to send it, right? Which is kind of the same as downloading in normal speak. And you want the complete record, and you want to hit file, right? Because you want to download it as a file, and then you want to create file. And then depending on how fast your computer is and your internet connection, which mine isn't, at some point. Maybe it's because of everybody here accessing GenBank at the same time. Anyway, that will bring you up a folder where you can save that. So label it something. Where did I save this for? Oh, I saved it here. I just, you see, I actually did it yesterday. Huh, how about that? I save it as yellow GenBank because that highlights the fact that it's the GenBank sequence. I'm not going to save it because I've already done it. So I'm just going to pretend that I did and go out of there. So now we want to actually look at that sequence, right? So this is going to be opening up. What was question 8? Whoops. Oh, there you go. I mean, that helped you answer question 8. So now we go to Eugene, which I've got open here. I'm going to, you know what pains me as much as it does, I'm going to get rid of that. And so what you want to do is just go to File, Open, or Control O. Where are we at? We want to go back to this folder. And yellow GenBank, here we go. You want to just open that up. And you can do some of the things that Dr. Page does to make it a little easier to visualize. I mean, it's up to you. I don't really mind. I tend not to bother. So essentially what you see here is, here's the genomic region here, labeled Y. Yeah, that's the yellow gene. This is your mRNA. Okay, some people have had issues downloading Eugene. Sometimes it just takes a while. If it causes you problems, so you can't complete it on time by tomorrow night's deadline, just let me know and I'll extend it for you. It's not a big deal. I think it just, depending on your Wi-Fi connection, it can take a little while to get it. You know, because it's a pretty full-featured program. I can't remember how long it took me when I did it. I guess it just depends. So again, this is essentially the same information that you had before, just in a little bit more detail. So genomic region, mRNA, right? And there's a big gap here, obviously, because there's an intron there. And this is the mRNA from 1 to 4,700 or something, I think, right? So exon 1, exon 2. Now your coding sequence, where actually codes for protein, begins here, right? So that's what, oh look, it's told us the answer again. That's what we're concerned with. And you can actually find that if you, I've got to move some things out of the way. Crab sticks. All right, let's move this down here. And this. All right, so the pale blue matches up to here. This is your exon 1, right? And you'll need to refer back to this when to answer another question, typically about, I think it's about what did we amplify in our PCR, right? So this is the end of exon 1. Let's scroll up a little bit and we'll find the start of exon 1. Come on. All right, so here is the start of exon 1. This is the mutant sequence. This is the yellow 1 allele sequence, right? Get off of that. Right, so things pop up so often, it's a little tricky to actually point at anything. So this is your coding sequence, right? This is your start codon, or what would be your start codon if you hadn't, or if there wasn't a mutation, a point mutation, changing this A to a C, right? So that is, if this is 170, this is 171, this is 172. Again, the same information you saw earlier on the NCBI GenBank page. If you click on this, I think it'll also give you the coding sequence, right? So this goes from 172 to 409. You can always make a note of that when it comes to answering some of the questions later as well. Okay, so you can just like kind of browse around, you know, have a little look, that kind of stuff. Okay, so thank you, Ali. Let's see what's next on the list of things to do. I wish I had a bigger screen, make my life a lot easier. But then again, it'll probably make my legs ache having it on my lap, so. Okay, So, what was question 9 again? Let's get back up to. Oops, too far. Majority of positions with noisy sequences are located at the ends of the sequence. And this is especially true of the beginning left-hand portion of the sequence, right? So if we just go and open one of these, if you'll let me. Nope. I don't know why this is such a pain in the ass sometimes. That didn't help. Text editor then? No, that didn't help either. All right, let's just get rid of these. Make my life a lot easier. All right, so let's open one of the yellow files. Damn it, why does it not have the sequence information in here? This is such a pain. No. I don't know. Sometimes Eugene gives me absolutely fits, I have to say. Come on, anytime today. See if this works any better. I don't know. Does anybody else have this problem?
There's a zoom button right there to the left of the sequence. See it?
Oh, right. Okay. There you go. Thank you.
No problem.
Why it would not go to that in the first place? Who knows? Anyway, so this is an electropherogram, right? So this is the actual peaks of the fluorophores, right, as they come out of the capillary sequencer. And what you tend to find is that it takes a little while for the process to get going, right? So the primer, well, there's no point me pointing it on the screen. The primer that you're using to do this sequencing is over here, kind of where this text box is, right? So the very first fragments that come off of that, the really shortest ones, tend not to give a very good accurate noise signal. And so what you tend to find is that the very first bases will give you Ns, and N means any nucleotide, right? So it's basically not able to call whether this is a ACG or T. This one here is a little odd, but you can also see here, this is like 1 peak, but it calls it as 2, right? So it's not really until about 30 bases downstream that you really get into good quality sequence. And if you scroll all the way over here, we don't actually have a fragment long enough, but what you tend to find is that the accuracy then drops off at the end of the sequence run. And so this is actually well within capillary sequencing kind of limits, which is typically like 700 to 800 base pairs, give or take. But you will find that the background noise, right, the bottom of the peak gets higher and higher as you go through, and then essentially it gets less and less accurate. So because the sequences that we're working with, the fragment is actually very small, you won't see that on the right-hand side, on the 3' end, but you certainly will see it at the 5' end, the left-hand side of the sequence. Oh, okay. Ha! I should have done what the instructions said, shouldn't I? That would have been a good idea. No, get away. Okay, so basically to be able to do a good mapping, you really need to get rid of this junk, right? So at the very least, you've got to get rid of any ends. Otherwise, when you map this, you'll find that the mapping isn't super accurate. So I think the next one is, yeah, there we go. Mapping our sequences to a reference sequence. So to do this, you need the reference sequence. Actually, no, I don't think we do. Scratch that. So tools. So you go up here, toolbar, Sanger data analysis. There. Map reads to reference, right? Those are the things to do. This one. Yeah, you can also put ends on the noise. To be honest, typically whenever I do any of this kind of analysis, I just chop off everything up to 30 base pairs. You always design the primers to cover that and more, right? So that's also another thing you can do if you wish. So the primers, you never design the primers for your PCR or for the sequencing as well, essentially. But you never design them so they're within 30 base pairs of the thing that you're interested in, because you know that those first few bases will be crap. So you just take that into account in the design, right? So when you want to do the, well, because it's doing it properly, right? So once you do this a few times, you learn to take these shortcuts, right? Which through experience, I know are fine. But you're not necessarily going to have that experience yet to make those kinds of shortcuts. So you can, yeah, just take it from me. You can do that. It's easy. All right. So we want to get yellow GenBank. That's going to be our reference sequence. And I'm just going to grab a bunch. There you go. That'll be enough. You're supposed to select all of them because that's the proper way of doing things. I'm just going to do a little bit faster and skip doing all of that. So then you just do map, the button at the bottom, and it will trundle away itself. You got a little process bar at the bottom. Quit it. Okay, so this is our mapping, and basically it's, you want to make sure that it fits, you know, like the sequence that you, or bit of DNA that you have sequenced is actually something to do with the yellow gene, right? It's not some random ass piece of E. coli genome. And so the bit along the top here is, or along the top here as well, is our reference sequence. That was what we downloaded from GenBank. And down here, these are where all of our reads, the sequencing reads, that's where they map. And so that little gray box basically tells you that's where the cursor is at the moment. So we can scroll over. Let's make this a little bit smaller. Right? And we should be getting some hits. So the primers, again, are designed to be a little bit over that away. Right? And so the primer is designed to the 5' UTR. because we know that the start codon, well, maybe I told you an answer there. The start codon is what we're interested in, right? So we know that the primer has to be in the 5' UTR, you know, at least 50 bases upstream, right, of our region of interest. And this is probably around, you know, what's that, 70? Okay, now because I'm a lazy ass, I didn't chop these off. So we get a real nasty, messy mapping. This is why you need to get rid of all that junk, basically, because otherwise it produces a pretty crappy consensus sequence. However, what the hell is an N in there for? This is base 172, right? So this is the first base of the start codon. We know that in our reference sequence, which is the yellow mutant, that's a C, right? So I don't know what this N is doing here. Maybe some weird ass crap is going on with this sequence. Yeah, that looks just like a bad call, to be honest. You see how it's thinking that there's a base there and there isn't? This CTG is the start codon, or what would have been the start codon in the wild type sequence. So you can see this read, right? Whoever AFCG were, no idea. This is a wild type sequence, right? So this must have come from a brown bodied fly because we have a proper start codon here. This is ATG. Here, we don't. This is CTG. So this fly, whatever that was way back whenever those sequences were generated, this fly must have been a yellow fly. Now, if you have a heterozygote, then you will have essentially an A and a C in this position, right? Because you'll have one copy of each. So even just by looking at this, looking at the electropherogram, you can actually tell whether or not that fly is homozygous dominant, homozygous recessive, or heterozygous. Obviously, if it's homozygous anything, it'll be one letter, dominant, or another, recessive. But if you have both letters present, and you'd probably get like some, oh, actually, there you go, you get an M. M stands for A's or C's. I didn't know that. And actually, if you hover over it, you'll see that a little bit over half of the sequences that I just randomly picked are from yellow flies, because 58% of them have C, where they should have an A. So that's also pretty cool. I actually just accidentally hovered over that and found out that information, which is kind of neat. This N, or here in the consensus, is because of this funky sequence here. And do we have any others? No, it's just that one. So if it were me, and I was trying to do this like proper, I would just say, oh, that sequence sucks, and can it? I would just get rid of it. And that would tidy this this mapping up a whole lot better. So which one is that? BLST. I'll remember not to use that one in future because it just makes a mess. All right, so so without that one, you would not have this dash, and this dash is basically a way of saying there's an extra letter here somewhere that is not shared by all of the sequences, right? So every place where you have a letter here, right, it's because that's a letter shared amongst all of the sequences. Where you have a dash, that's because one of these sequences is crap. I wonder if I can just get rid of that. Ooh, maybe I can. Did that work? No, it didn't. Oh well. Oh yeah, there we go. Doesn't fix the consensus at the top, but never mind. No, dash is a artificial space introduced into the alignment to deal with a sequence that has one too many nucleotides. So when you have an issue reading the what base is present, actually I think it trimmed it out for us automatically. Yeah, it did. Oh no, there's still one here. When you have an N, that means that it could be any nucleotide, that there was a nucleotide there, but it couldn't tell which one it was. And usually that's because the level of, yeah, so here, for example, you've got green, which is A, and is it T is red? Yeah, oops, make sure I get that back in the right place. And so you have an equal level of red and green fluorophore, so you can't tell whether it's an A or a T. And so it just goes, yeah, don't know, and puts in an N instead. For any is essentially what it means. The dashes are more artifacts of the alignment. So, for example, there's a dash here, which is because somewhere in here, there's an extra, one of these reads has an extra T. I'm not entirely sure why that dash is there actually. Anyway, maybe that was caused by the other sequence. Basically, sometimes you just get some shitty sequences and they screw things up. And so usually you would take those out because they're just a royal pain in the backside. Other times you can actually edit them, go and actually look at the electropherogram and go, no, I don't think that's right, and cut it out yourself. We don't need to go into that kind of detail here because it lines up pretty nicely. Right, so that's the, what other questions were there for the mapping? 10 to 13, what are the questions for that? Okay, so given what we know about what a start codon is, and what we've been told by the flybase gene information, you should be able to answer question 11, right? So we're dealing with the mutant sequence, which has a CTG. The wild type has an ATG, so you should be able to answer question 11 from that. So given what question 12 is fairly straightforward too, right? Think about what the AUG is involved in and what would happen if you don't have a start codon in terms of gene expression. So that will help you answer that question. And 13, which components of the yellow gene? Well, that you go back. Actually, where would I go to have a look at that? Okay, so let's go back to this version of Eugene. Eugene. I'm sure that's a song of some sort. Okay, so where's our sequence read end? Oh, crap, I've got this in the way. 375. Jolene. Oh, there you go. Almost. 375. So now we just go back to this and say, okay, what is covered up to 375, right? We've got a big chunk of exon 1, and we have our 5' UTR. And let me just double check the test to make sure that is actually correct. I might have to edit the because that's actually, hang on, let me just hang on before I do anything else. Let's have another look at this again. I don't want to go fiddle farting around with. Okay, so where does this map sequence map? Ooh, gone too far. Map to. Okay, so all right, because it's not the length of the sequence that matters. It matters to what it maps to. Okay, so let's get out of that so you don't see that by accident. Let's go back to my screen. Okay, so going back to the map or mapping. Let's bring up chat again, if it lets me. Where be the chat? Fuck nuts. Chat's disappeared. Ooh, pardon my French. Anyway, zoom. These are our sequence reads. So you just need to scroll along the mapping. Oh, here we go. We've got some crappy sequence popping in over here on the side. And if you were to take this conservatively, that would go to, let's say, 445 base pairs, right? In terms of the reference sequence. So now go back to your reference sequence, which I've got over here. And you can see that, if we just scroll down a little bit, 445 is about here, right? Which, or if you can see where I'm pointing at on the screen, it's part of Intron 1. So that tells you that our PCR product covers everything from the left, which you know what that is, through the middle, you know what that is, to the right, which is where I'm pointing, and you know what that is, right? So if you look at that, you should be able to answer that question just fine. Okay, now let's see if I can get my chat back. All right, so the next bit is, stick that over there, bring this back. Is everybody keeping up so far? It's a bit of a, sometimes I go a little bit too fast just because, you know, I kind of know what I'm doing essentially, or sort of. Sometimes I know what I'm doing. So say, oh, I can't get the chat back. Where the hell has that gone? Blind squirrel finds a nut every so often. Okay, so now what we need to do is do an alignment. So an alignment is essentially a fancier version of a mapping, right? And so now we can have both the wild type sequence and we could have the mutant genomic sequence from GenBank if we wished. And we can put in all of the sequence files that we also have. So let's go do that. This will actually take a little while to do. So I'm just gonna, let's see if I can actually open one I did before. Let's see. Okay, so I'm gonna open a whole bunch of these. So I'm gonna select all of those and get rid of that one because that one's a turd. We don't want that. And then slowly these will all open up. Do I have here? I don't think I do. So let's open up my GenBank sequence. Get that open too. And then we go here, and essentially what you want to do is just highlight all of the ones that you want, and then select them all. Actually, you know what I'm going to do? I'm going to do a whole lot less because it will take forever and a day otherwise. So let's use this one, which I already have open. Let's just do these ones and get rid of that one again. Okay, so hold down Control and select all of these. And while you still have Control held down, right click or double click, whatever thing you use. That will bring up a little menu. Go to Export Import. That will bring up some more options, and then export as an alignment, right? And you can select that's what you call it. Let's just do a mini alignment. Okay, so this looks like crap because we've got all of this junk still here. So you go through those, and you just select them from here to here. If you don't, it will produce a horrible alignment, right? Because the computer will try and figure out how to fit all those ends into the alignment, and it just won't work very well, right? So you need to have good quality sequence data to get a good quality alignment out at the end of it. Oops. Let's just do that. Okay, and then just go back to this because I'm not super familiar with this. All right. Then you go to the actual window that you're in, click on the top, that's your reference sequence, and then hold down Control and click on the bottom one, and then right click again while holding down the control panel. And then you go down to Align, right, which is to do alignments. And essentially all of these are different ways of doing alignments, and we're going to use the Cluster W alignment option, which is that one. Click on that. These are a bunch of settings which you don't need to change, but they're essentially what you would tweak if you wanted to fiddle around with the alignment. If it gave you like some really crazy options or output, then you would fiddle around with these to try and get rid of that. Essentially, say you're working with like divergent species sequences, and it was struggling to match them up as they should be matched up, as you know they should be, then you can fiddle around with these to try and get that alignment looking better. Also, we're not going to do it here, and I'm pretty sure you can do it with this program. I'm not 100% sure. But these alignments are also what you would use to create a phylogenetic tree. So obviously, we're not going to do that in this case because these are all from the same species. So, you know, I mean, they're all the same. But if you were, for example, make an alignment of the yellow sequences from all Drosophila species that have been sequenced, and if you're bored, you know, you can always do that for fun. If you were to do that, then from that alignment, then you could construct a phylogenetic tree, which would give you, according to the yellow sequence at least, evolutionary relationships between those different species. So if you were to take this one step further into the more kind of systematics or evolution aspect, that would be the next step after doing this alignment. So we're just going to click on that, and this takes a little while. So now's a good time to ask questions. Oh yeah, that's, I mean, the thing is Dr. Page does everything like correctly. Because he's very, you know, he's very good about doing things the right way, and it's good to teach people to do things the right way. I'm a little sloppier, so I'll do things the quicker way, and you can just delete them off of the sequence itself. So that's more of a kind of stylistic thing, I guess. So this is the alignment, right? And down here you can see essentially which area has the highest alignment. And it's telling you it's right at the beginning, which is good because that matches what you already know. Let's move this over a little bit. Make this a little bit smaller too. Okay, so you're gonna scroll over here. Okay, so now, right, we've got some crufty muff down here, like some jittery nonsense. And so we're not really sure about this part. This is just junk. But essentially, these bars at the top tell you how many of your reads or your sequences of interest, right, i.e. not the reference sequence, have the same base as the reference sequence. So if you were, for example, doing this between different yellow genes of different species, you would expect those bars to kind of increase and decrease depending on how conserved the residue was at that particular position. And you tend to find like clumps of conservation between different species. But we're not doing that. We're just doing it between different genotypes. And so now, ta-da, this is what we see. And so this now tells us, I think that's, oh yeah, it's even higher just because I chose different sequences, right? In this case, because I only chose a subset, 6 of them out of 30 or so, 71% of them have the point mutation that's in the mutant, in the yellow 1 mutant. 29% of them have the wild type A for ATG, right? So this one here, oh, bollocks. What have I done? Go back, back. Oh, I don't know what I've done. Sometimes these things are more powerful than they really truly need to be. Let's go back over this way. I really truly don't know what I did there. Phew. Back to where we should be. So you can see here, this is your ATG, this is your start codon. So obviously when transcribed, that would be AUG, be recognized by the tRNA methionine, and translation would start. All of these other ones are from yellow-bodied flies because they have a C instead. And so this would transcribe into CUG, which is not translatable, right? So you would not get any protein from a fly that is homozygous recessive for this allele. And that's pretty much it. Where's the chat? Chat. Ah, crab sticks. Well, does anybody have any questions about this right now? Hopefully, between this and the videos that Dr. Page made and I still can't get my chat back. So random. No. Between this, the videos that Dr. Page made, and what we've done here today, it should be a lot clearer. This isn't due tonight, it's due tomorrow night on Wednesday. There you go. Wednesday the 29th. So if you still have some trouble, I'm going to be, online tomorrow at various points, and it's not due until midnight. So if you do have any issues, just give me a shout, and we can go through it from there. So again, a lot of this, I think most of the confusion is sometimes like there's a little bit of assumed information that, because we've done this so much, we assume that and those little bits, because it's fairly particular about how you need to do stuff. Those little bits can get in the way and make things not work. So hopefully, having seen it being done, those little miscommunications or gaps in understanding are going to be filled. And so it should be a lot easier to do. And that's pretty much it. Yes, Angela, I did. Does anybody have any questions about the assignment or even about any of the material we're going to be on the exam? I still have at least 20 minutes before my coffee is due. The files that are downloaded, is that just in general or in UGene? In UGene. Okay, so hang on, Olivia, just give me a second and I'm answering another question. So this was a kind of a mistake that I made. So let's click on, hang on, let me get this sharing back up again. So if you double click on that, okay, activate view. So my mistake, which Ali and Angela cleared up, is you see the fully zoomed out version of the electropharogram of the sequence. So if you can see that tiny little magnifying glass, it's actually so small you could do with a magnifying glass to see it, right? If you hold over that and zoom in a little bit, then you should see the sequence, right? And you can kind of figure out what degree of zoom kind of works best for you. So once you've opened up the sequence in Eugene, which you just do by going to File, Open, and then select whatever folder you have these sequences in. Just click on one of these. Let's say this one. Right. That will then open that up. See, it's down here underneath the alignment I just did. And then you just need to click on the zoom button. There are all kinds of funky things here. I don't know, you can get rid of all the A's. Don't know why you'd want to, but you can if you wish. What does this do? Quality bars. There's so many different bits and pieces in here that I just don't even know of. I could spend hours just wandering around looking at stuff. But that should allow you to see those files. Did that help? Sorry, this is a question to the one person that asked me. Yeah. And in the meantime, everybody else has answered Olivia's question. Yes, the exam due for midnight tonight is, oh, good. I'm glad it did. Is the last one of the course. So this is it. There is no more, which it could be both a good thing and a bad thing, kind of depending on your point of view. Yeah. And again, it's not cumulative. It's just, which probably by this point is not going to make any difference, right? But it is on 17 and 18, which are gene expression in eukaryotes and prokaryotes, and 20, which is transgenics, which we covered today. Cool beans. All right, anybody have any questions? No? Well, it has been a pleasure, I have to say. It's been a very interactive and enjoyable class to teach. So thank you all for all of your contributions to this class. It really did make a difference. And good luck on the exam. And yeah, hopefully I'll see some of you actually in person someday. That'd be kind of neat. I imagine that you'll recognize me unless I shave and go incognito. So yeah, have a wonderful rest of your summer. Good luck on the exam tonight, and let me know if you have any problems either with the exam or with finishing this lab due tomorrow night. Yeah, pre-quarantine beard. Anyway, I'll post all of this stuff up as soon as the files have finished converting, and you'll be able to kind of go back and reference any of that if you need to for that. And again, Angela and anybody else, think about what I offered, and if you do need that, just let me know cryptically. Cool. All right. Catch you later, everybody.